सगळ्यांना पीपीटी दिसते का? Can everyone see the PPT? कविता,
हाँ सर.
हा पीपीटी दिसते का सगळ्यांना?
हो सर.
So we'll study our next point, that is phytochemicals in
सर
health.
तुमचा आवाज येत नाही.
आवाज येत नाहीये? हॅलो,
हा लय हळू येतोय. हॅलो.
हा ठीक आहे, मी जोरात बोलतो. So we'll study
हा.
phytochemicals in the healthcare. So these phytochemicals, if we study, a lot of Ayurvedic plants we are using. So India is very famous for these Ayurvedic, Indian Ayurvedic drugs. So which drugs there are? There are like Taxol. Taxol is used for treatment of cancer. विनक्रिस्टिन आहे, विनब्लास्टिन आहे, कॉल्चिसिन आहे, आर्टिमिसिन आहे. आर्टिमिसिन इज ऑल्सो चायनीज anti-malarial drug. This species also is found in our Indian subcontinent also. So there are around 25,000 flowering plants in worldwide, and out of this only 10% have been investigated for medicinal properties. So what in bioinformatics they are doing? They are doing that is they are maintaining the database. What they will do? They will survey the medicinal plants. They will link it with the taxonomic status, and if we are getting a species which is closely related, for example, to Artemisia, which is producing artemisinin, so they can estimate which species artemisinin is present, the close member to this Artemisia. So they will screen this. By this, they can convert the database, and it will be easy for us to screen these drugs. Then coastal agriculture limitations. So coastal region, what are the characteristics of coastal region? You know, coastal region कुठल्या ह्याला म्हणतात? Coastal region. Can anyone tell? What is coastal region in India? Where we have got coastal region? Hello. Rajkor. Rajkor.
सर नाही माहित.
कोणाला माहिती आहे का? Coastal region कोणाला माहिती आहे का? Which part is known as coastal region?
No, sir.
The area which is nearby sea. The area which is nearby sea is called as coastal region. So salinity is the major problem. Why? There is floods are there, or there are some backwaters are there, or there is tsunami. During this, what happens? The sea water enters in the land area. When it will enter in the land area, what will happen? Sea water is saline. So salination will take place. Due to salination, what will happen? What happens? The salt level in the soil will increase. If the salts increase, what will happen? The plants cannot be grown. बरोबर आहे?
हो सर.
Yes, sir.
Ha. So a second reason is where overuse of this fertilizer is also causing increase in soil salinity. So increase in soil salinity is directly proportional to the decrease in crop productivity. So plant productivity will get decreased. It will lead to what happen? Income of the farmer will also get decreased. So this can be solved by how? How it can be solved? See in coastal area, if you study the plants, diversity of the coastal. सगळे beach वरती गेलेत का? Everyone has seen the beach. समुद्र
Yes,
किनारे
sir.
सगळे गेलेत का?
Oh,
So there you can see, if you are going to Mumbai on the highway, express highway near the sea, you will see some big plants are growing that are called as mangrove forest. So they can go dry directly in the saline water. So what is there? There they have got presence of genes which allow them to work in coastal environment. So what happens? Due to presence of these genes, what happens? Some biochemical reaction takes place, and due to this, what happens? The plant are able to survive in the coastal region. So scientists, what they are doing, they are isolating these genes, and they are transforming these genes into normal plants. For example, there are few species of rice which can grow in saline water also. They are found in Kerala region. But all varieties of rice are not cultivated in saline water. So what happens? You can do transformation of rice with the gene containing salt resistance. So what for transformation, what you have to do? You have to isolate the gene which is present for salinity, which can help against salinity. Now for isolating the gene, what you'll do? You'll design forward primer and reverse primer, and you will isolate a specific gene from n number of plants, different species of mangroves. And then what you will do? You will create a library. Now your kit, आपली library कशी असते पुस्तकांची, you can see it physically. But here what you do? You create this genetic library. How you are going to create? You are going to clone it into vector. In genetic engineering we have studied, and you will clone this and you will keep this into E. coli. And in this manner you will create thousands of clones of this E. coli. So while analyzing, what you'll do, you'll require the bioinformatics tools. What you'll do, you will put the sequence in that plaster sequence bioinformatics database, and there you will screen for the related protein. From that you will know which gene is present, name of the gene, their transcription factors or zip proteins. All this you can know from the DNA sequence. So here bioinformatics has the role for isolation of these novel genes. कळालं?
ah, sir.
So next part comes, that is bioremediation and environmental biotechnology. Now bioremediation, if you will define, it is a biological process for what happens, for converting toxic waste into non-toxic waste. So it is a very hot spot area now, this bioremediation. Why? Heavy industrialization has led to lot of pollution. Then if you see this human population in the cities, they will create lot of waste. Home waste is there, then sewage waste is there. They will use vehicles. This use of vehicles will lead to lot of air pollution. There is also sound pollution. So all this, it can be tackled by using living organisms. How? Can anyone give example of transgenic bacteria which is used for oil degradation? Who has worked this on this topic? सांगू शकता कोणी? Last time I've told you, mineral oil deposits are found in the Gulf region. So during this oil is transport from ships. So if there is any leakage, what will happen? The mineral oil will get deposited on the sea water. At the oil, what are the characteristics of oil? the oil will keep on floating.
हॅलो सर.
हा. हॅलो.
हॅलो.
हा, बोला.
हा सर, ऑइल स्पिल करणारा बॅक्टेरिया कोणता?
कुठला आहे सांगा ना. ऑइल स्पिल नाही, ऑइल डिग्रेड करणारा.
डिग्रेड करणार आहे.
इट इज सुडोमोनास.
हा
सुडोमोनास.
हा हा हा.
सो व्हॉट दे डीड की हे काय होतं, मिनरल ऑइल इट कीप्स ऑन फ्लोटिंग. त्यामुळे फ्लोटिंग असल्यामुळे काय होतं, देर इज लेस कॉन्टॅक्ट ऑफ सी वॉटर सरफेस अँड एअर. So what will happen, the oxygen will not get dissolved in the water. मग खाली जे पाण्यातले ऑर्गॅनिझम्स आहे, they will face oxygen deprivation environment. If there is no oxygen in the water, they will get dead. So a lot of fish will get dead, the birds will get dead, because if they will sit on that oily water, what will happen, the oil will get deposited on their feathers, so they can't fly, and whole water surface will get black color. So what they did, they isolated genes which can degrade this petroleum, and they created a transgenic vector, recombinant vector, and plasmid vector. And this plasmid vector, different type of plasmid, they expressed it into pseudomonas. And scientists, what they did, they sprayed the pseudomonas suspension on the seawater. So what happened, the pseudomonas started consuming this mineral oil. आता mineral oil काय, what is mineral oil, how mineral oil is formed, thousands or lakhs of years earlier, what happened, algae and other biomass was growing on the ocean. After that, what happened, they settled down, the atoms, and the soil got deposited. मग काय झालं, during course of time, what happened, there was no degradation of this organic matter, and this organic matter, due to high temperature and heavy pressure, they got converted into mineral oil. They're nothing but organic remains. So organic molecules, what they are consist of, proteins, carbohydrates, vitamins, fats. आपली बॉडी कशाची बनलेली, ह्याचीच बनलेली आहे ना, and water. So what they will contain, major what, nitrogen, carbon and oxygen. Carbohydrate will contain what, cyclic glucose molecules. Fats will contain transistor. Proteins will contain amino acid. Amino acid contain what, nitrogen, carbon and hydrogen. ह्याचेच बनलेले आहेत ना. So what will happen when they will degrade this? So petroleum is also made up of all these compounds. फक्त त्यांची अरेंजमेंट वेगळी वेगळी. पेट्रोलमध्ये सायक्लिक अरेंजमेंट आहे, डिझेलमध्ये वेगळी अरेंजमेंट आहे, गॅसमध्ये आहे ते वेगळे अरेंजमेंट आहे, ब्युटेन आहे. So what this organism will do, they will cut this cyclic ring, and they will utilize the energy after oxidation of these compounds, and they will grow, and they will convert it into CO2, NO2, water. आलं लक्षात? हॅलो.
हो सर.
So they have converted this pollution, they have decreased this pollution by using this transgenic pseudomonas. So you want to isolate the genes. Here again comes bioinformatics application. You have to do the forward primer, reverse primer. Then you have to clone that in the vector. Then you should know the genetic map. How you will know the genetic map? You will cut it with the endonucleases. So when you will cut it with endonucleases, you will get small fragments. Then you will go for sequencing of this. When you know the sequence, then you will know the ATGC sequence after sequencing, and then you store this data in a database, and then you can take and use that database for designing new genes. So in this matter, this bioinformatics is very important here. Others are fungi. How fungi are reducing this pollution? So whatever waste you are producing, fungi, they will contain what, cellulose enzyme they have got, fungi. So this enzyme will what happen? It will degrade this plant waste and convert it into a compost. So lot of fungi they are used. Also fungi used for degrading, fungi and bacteria used for degrading dyes. Then third are plants. How plants they are using helping. Plant will take complex organic molecules. Sorry, plant will take what? Simple organic molecules which are degraded by fungi and bacteria, and they will use it and they will grow on that compost. So there are a lot of plants which can show also dye degradation ability, insecticide degrading ability. They will absorb heavy metals from the soil, and they will decrease the pollution level. They will absorb CO2, and they will provide oxygen. So you got how bacteria, fungi, and plants are used for bioremediation, and how bioinformatics plays a role. कळालं?
हाँ सर.
The next is structural biology. So first we do research, then we'll get what you'll use physicochemical, molecular, and computational methods you'll use. Then you will design the structure of biological molecular molecules. There, when you know the structure, you will know their functions, and you will use this what you will use where in pharmaceutical, food, agriculture, and other biotechnological applications. How are you going to use it? Last time I've told you, if you are getting a new drug, you want to design a drug for, say, COVID. So what you'll do, you will take this virus molecule, virus DNA or RNA, you sequence it. From that, you will know the proteins. From that amino acid sequence, you can know from that amino acid which protein is developed. You can know. Then you can virtually create the 3D structure of this protein, and you will study the interaction of thousands of drugs at a time with this molecule. That is soft lab experiment you will perform, and out of that you will get few promising drugs, and then you will go on further for the trials. So they will ease the time period required. So bioinformatics has a very important role in structural biology. I've given example of COVID. There are many other examples in food, agriculture. Agriculture मध्ये असेल, you want to develop a drug for insect, for killing the insect. Pest is there. Then you have to develop these biosensors for water stress level in the plant, or attack of any fungal disease. There also you can use this structural biology, because everything fungi, how it will degrade the plant by secreting hormones, enzymes, and this enzyme will convert our plant biomass into useful product for fungi. For example, cellulose is there. Fungi will secrete cellulase enzyme, and the cellulase enzyme will degrade the cellulose in the plants, and fungi can utilize the glucose. In this matter, fungi will infect the agricultural crops. So in this structural biology, you can use various bioinformatics tools. Then comes genetic engineering. Now I don't have to tell you separately where genetic engineering is used. Pharmaceutical मध्ये example आहे insulin production in the E. coli. All our antibiotics, drugs, everywhere this genetic engineering is used. Then gene therapy. Gene therapy, you can, for example, if the parent have any genetic disorder, we can treat this genetic disorder by using gene therapy. Then is you will study this in animal biotechnology in depth. Gene therapy. Then transgenic plant, BT cotton is there. Transgenic animals are there. So they are converting a lot of fishes. Transgenic fishes are there. Their cows they are producing transgenic. Sheep they are producing transgenic, and transgenic other microorganisms, bacteria, algae, fungi, viruses they are creating transgenic for using as bacteriophage. So these are applications of genetic engineering. We have studied in detail in this genetic engineering. In genetic engineering subject, we have studied this in detail. Then biotechnology applications in diagnostics. First it will go identifying organisms. How you will identify the organism? how you will identify bacteria? Can anyone tell? By using molecular methods, how you identify the bacteria? सांगू शकता कुणी? संग्राम?
बाय द सिक्वेन्सिंग म्हणजे स्ट्रक्चर त्यांचे काढून
Which sequence we take? RNA ऐकलाय?
हा सर.
In bacteria 16S RNA is present, and in eukaryotic there is 18S RNA. So for 16S RNA, what they have got, forward and reverse primer is there. So whenever for example, I am working on a bacteria which is producing high amount of cellulases, I want to identify that bacteria. One method is do biochemical, morphological biochemical test. तुम्ही सगळ्यांनी केलेले आहेत. ग्राम स्टेनिंग केलेलं आहे, मोटिलिटी केलेली आहे, कॉलनी कॅरेक्टर्स बघितलेले आहेत. Then you use Burgess manual, and second is molecular biology concept. I will give this bacteria to a laboratory which is doing bioinformatics analysis. What they will do? They will take this bacterial colony and they will do PCR by using forward and reverse primer for 16S RNA. So this is a conserved region. By knowing this, they will get amplified this gene for 16S RNA, and they will put this amplified product, PCR मध्ये काय होतं, amplification होतं DNA चं, in the cloning vector. And in this vector what they will do after you have put this into cloning vector, they will do the sequencing of that DNA. Forward and primer run करून ते sequencing करतील. Once you have did the sequencing, you will know the sequence ATGC. That sequence you'll put in a bioinformatics tools. And what you'll do, you'll find that sequence for any similarity with other sequences in biological database. And here you can know exact how much percentage these organisms match with other sequences. If the percentage is less, then you have got a novel bacteria. You can know the group. And if it is more similar, you can say this is similar to this bacteria, Cellomonas for example. So all this comparison is done by bioinformatics tool, and we can get the name that service provider will give the name of that organism. What you don't have to do colony characters, biochemical test, nothing. Only you have to give a pure culture of that organism on slant for sequencing. And from that sequence analysis, 16S RNA, you can do identification. Same goes for algae, fungi, which are eukaryotic. त्यांच्यामध्ये काय असतो, 18S RNA असतो. Then second part comes identifying genes. So here you can also identify the genes. But gene identification, same this process. Sequencing वरनं तुम्ही genes identify करता. If you are studying, you are doing transformation in the plant and you want genes which are resistant to salinity, you will identify these genes from saline plants. Then third is diagnostic, infection, diseases and genetic disorders. So if you are going to study, for example, if the parents have diabetes and you want to study whether that baby has got diabetes or no, it will get. What you will do? Specific markers are there for diabetes or cancer. If the parent have got cancer, you can take this forward reverse primer and you can do PCR. And from this, you can do sequencing of the DNA in that baby, particular gene. And from that, you will know whether the baby will develop cancer or diabetes. So it will help in health also. So people who are doing, they are called as genetic engineers in biotechnology. So you can engineering से काम क्या है, build and develop new techniques. So same here we are doing in genetic engineering. Then as usual, molecular biology. What is this molecular biology? So you use molecular biology, microbiology, biochemistry, genetics, chemical engineering, cell biology. All the techniques you'll use and you will develop drugs, vaccine, new crops with new abilities, diagnostic tools आहे, and then livestock. Under this livestock, you'll get cow, buffalo, fish, poultry, all the animals which we are rearing for our use, silkworm, it comes under livestock. So like livestock improvement. The microbiology and its uses. So what microbes will consist of? Small viruses, bacteria, and other unicellular organisms. So if you'll study them in detail, you'll get various antibiotics, food products. So food products where microbiology is used is idli. Here you are employing lactic acid and yeast. Second food product is where microorganisms are used. Can anyone give example? कुठं वापरते फूड मध्ये मायक्रोब्स कुठं वापरतो आपण दुसरीकडे फूड मध्ये? कोणी एक्झाम्पल देऊ शकतं का? राजकुमार, हॅलो,
हा सर.
एक्झाम्पल सांगा ना, फूड मध्ये कुठं वापरलं जातं? अरे वेर यू आर युझिंग मायक्रोब्स इन फूड? Bakery products, bread. What you are using here? Yeast. While preparing curd, you are using lactobacillus. So you have got many probiotic bacteria also, which are used for human and animal welfare. They will contain lactobacillus and other species. Other where you are using whole wine industry or
Yes.
alcohol industry, it is using microbiology only, na?
Yes.
Then have you have Chinese food? In that Chinese food, you are using soya sauce.
Yes, sir.
So it is also fungus. So there are a lot of microorganisms which are used for food industry, similar for pharmaceutical industry. Mata, here comes the role of microbial cultures. So if I'm developing a new strain of, say, give you example of cellulitic bacteria. What I will do, I've isolated a bacteria which is giving very nice activity of cellulase enzyme, and it can be help for producing or degrading this organic waste. What I will do, I will take that bacteria, I will sequence it. I've got a novel bacteria, I have got rights to give a name to it. I will name that bacteria and I will deposit it into a culture collection center. लक्षात ठेवा, culture collection centers. So what they will give, they will give a accession number to my culture, and they will store this culture in their culture collection center by using freeze drying. Freeze drying मध्ये काय करतात? The bacteria is grown in a broth, and this broth is subjected to a freeze drying. म्हणजे काय होणार? All the liquid is removed and we'll get a powder of bacteria. This powder of bacteria is stored in the ampules. आपल्याला इंजेक्शनचे अँप्युल्स मिळतात, त्या सेम इन दॅट सेम मॅटर they will store this bacterial powder in the ampules, and these ampules they will maintain in -70 degrees Celsius. So for hundreds of years we can store this bacteria with their character in the lyophilized form. त्याला म्हणतात lyophilization. So in this matter we will isolate thousands of organisms. आपल्या इथं कुठे माहिती कल्चर कलेक्शन? इन द एनसीएल. इट इज कॉल्ड ॲट NCIM. आणि इंडियामधली लार्जेस्ट कलेक्शन सेंटर आहे चंदीगडमध्ये, MTCC. चंदीगडमध्ये मायक्रोबायोलॉजी इन्स्टिट्यूट आहे, तिथं लार्ज मोठ्या प्रमाणामध्ये हे मायक्रोबियल कल्चर्स आर स्टोर्ड.
Okay.
So for example in our MSc research what we want to do, you want to study the lactic acid fermentation. मग आपण बॅक्टेरिया आयसोलेट करणार का? आयसोलेशन मध्ये आपण 6 महिने घालवणार. आपल्याला वेळ आहे का 6 महिने? तुमचे 2 महिने तर गेले. प्रोजेक्ट तुम्हाला किती असतो? 2 ते 3 महिन्याचा असतो,
हा सर.
4 महिन्याचा असतो. what you will do, you'll readily order these microbes from that culture collection center. For example, if you want to study agrobacterium transformation, so you want culture of agrobacterium tumefaciens, so you'll directly order this, place the order, and they will pay the, you have to pay the charges, and they will send you the agrobacterium culture. For example, any industry is there, if any industry they have to produce citric acid fermentation aspergillus वापरतात, so they will readily take this aspergillus. Sugar industries, they want to produce wine, they will take yeast. They've got thousands of yeast varieties. Out of this you can identify which yeast has got good production, and you will order that yeast. फुकट देतात का ते नाही. For last time I have studied for NCIM, for research purpose they are taking 1800 per culture, 1800 रुपये, and for industrial purposes they are charging 5000. For MTCC Chandigarh, they are charging 15,000 for commercial applications per culture, and for research they are charging 5,000. So here also they are creating databases. They are doing PCR. These culture collections they are doing PCR. PCR केल्यानंतर they are identifying the genes, 16S RNA, and from there they are doing identification. With culture they are also giving consultancy for identification. So here bioinformatics is used. वर्ल्ड मधली सगळ्यात मोठी युटेक्स मध्ये आहे, युनिव्हर्सिटी ऑफ टेक्सास USA मध्ये मोठं कलेक्शन सेंटर आहे. तिथे दे आर कलेक्टिंग अल्गी, फंजाय, यीस्ट, बॅक्टेरिया, अँड यू कॅन ऑर्डर इट फ्रॉम युटेक्स, बट द कॉस्ट इज व्हेरी हाय. आलं लक्षात मायक्रोबियल कल्चर्स? कळालं का सगळ्यांना मायक्रोबियल कल्चर्स?
हा
हो
सर.
सर.
For example, some cultures are there, they will, what they will do, they will not give any. For example, champagne, if you consider champagne is produced where? In France. There is a town, the name of there is champagne. The champagne, what happens, the sparkling wines, when you'll open the bottle, there will be lot of face येतो. So they are sparkling wines, जर बघितलं असेल champagneची. So the climatic conditions there are very favorable. Soil condition is favorable, good quality of grapes is produced, and from that they will do fermentation and they will produce champagne. So champagne producing yeast, what happens, the mother culture they have subcultured and maintained this culture for 500-600 years. नेचर मध्ये आता मिळत नाहीत ते. So this type of important culture they will not submit in the culture collection. That industry will have its own culture collection, and they will not provide this strain to anyone. ते त्यांचं proprietary. आता ती इंडस्ट्री मोठी असेल सॅम्पेनला, त्यांना इट कॅन अफोर्ड to develop a whole cultural laboratory. But for example, take example, if I have got a novel culture, I don't have a culture laboratory, cellulitic. I will pay these charges to the culture collection to maintain my culture. माझी इंडस्ट्री आहे, if I am producing some certain kind of antibiotic, but I don't have a culture collection center. सिरमचे एक्झाम्पल घ्या आपण. सिरम मध्ये मोठ्या प्रमाणात वॅक्सिन्स तयार केलं जातं, आणि ह्या वॅक्सिन तयार केल्यानंतर काय केलं जातं, it is produced by upstream processes, and by downstream processes they will remove the, isolate the antibiotic. But the culture, if they got contaminated and they have lost all the culture, what will they do? कुठून आणणार परत तो strain? So they will deposit their culture in the culture collection center. This culture collection center, what it will do, it will take this bacterial culture, it will check its purity, it will grow the culture in the broth, and they will lyophilize it. Lyophilize करतील म्हणजे काय करणार, under -20°C using CO2 and freeze drying process, they will freeze dry the culture, आणि पावडर मिळेल तुम्हाला, आणि त्या पावडरला काय करतील ते, they will store it in the ampule, and they will store this ampule in the -80°C. Whenever a serum company is facing any problem with their cultures, they will readily place order in this culture collection. अशा पद्धतीने they will store n number of cultures, but they have got agreement with the culture collection center that they will provide this their culture to no other company because it is proprietary of Serum company. आलं लक्षात how this culture collection works?
हा सर.
ठीक आहे, मग आपण थांबायचं.
ओके सर.
कुणाला काय डाऊट?
सर बायोइन्फोची तुम्ही आम्हाला पीडीएफ सेंड करून द्या की.
अरे बायोइन्फॉर्मॅटिक्स आहे की नाही, सॉरी, बायोइन्फॉर्मॅटिक्स सरांनी हे केलेलं आहे.
पंकज सरांनी का?
हा,
नाही आता उद्या पेपर आहेत ना, म्हणून म्हटलं आपलं हे करायला.
बेसिक की नाही तुम्हाला हे केलेलं टीवायचं वगैरे बेसिक हे त्याच्यामध्ये असणारे बेसिक प्रश्न एवढे म्हणजे हे आहेत.
चालतं सर.
मी तुम्हाला जीईचं पाठवून देतो.
चालतं सर.
विशाखा विशाखा
हा सर.
हा,
सर.
आता तुम्हाला मागितले नोट्स मी विसरलो, आता लक्षात आलं, मी स्कॅन करून पाठवून देतो नोट्स तुमच्या ग्रुपवरती.
हा.
ओके सर.